Hello friends, we have already discussed about several DNA replication enzymes like DNA Helicase, primase, topoisomerase and DNA polymerase in separate videos. You can refer my videos, the link is given in the description box. Today we are going to discuss about DNA ligase enzyme. DNA ligase enzymes are also known as molecular glue. It ligates or joins two DNA fragments, sometimes also RNA, which are separated. It was first characterized by Wyss and Richardson in 1967. It joins the separated DNA chains by catalyzing the formation of phosphodiester bond between the 3' group at the end of one DNA chain and 5' phosphate group at the end of the other. This phosphodiester bond formation reaction requires energy source as it is thermodynamically uphill. So what are the energy sources? ATP in case of eukaryotes and archaea, NAD plus in bacteria. DNA ligase enzyme play a major role in DNA replication. Other roles are as repair enzyme, also in cloning and next generation DNA sequencing. This we will discuss in detail in separate videos. Today we will focus on DNA replication. In DNA replication, lagging strand is synthesized as Okazaki fragments. Primer of each Okazaki fragment is removed and replaced by DNA polymerase 1 enzyme. By the 5'3' exonuclease activity, it removes the primer and replace it with DNA sequences by 5'3' polymerase activity. And this is seen in prokaryotes. Removal of primer and its replacement with DNA sequences is different in eukaryotes, which we will discuss in eukaryotic DNA replication. But after the primer is removed and it is replaced by DNA sequences by DNA pol 1, nick is present between the DNA chains. Nick is actually discontinuity in DNA strand due to the lack of phosphodiester bond between adjacent nucleotides. And this nick is sealed by DNA ligase enzyme. This is the parental strand and this is the lagging strand. It is present in the form of fragments known as Okazaki fragments. And these are the primers of Okazaki fragments. Now DNA polymerase 1 enzyme comes and removes the primer and replace it with DNA sequences, but nick is present between the DNA chains. Then comes the DNA ligase, which seals or ligates the DNA chains by forming phosphodiester bond. Now complete strand is formed. So let us discuss in brief the catalytic events that occurs during ligation. ATP comes and binds with the ligase enzyme. Then inorganic phosphate or pyrophosphate is released and covalent bond forms between ligase and AMP, adenosine monophosphate. Now the ligase enzyme is activated. This activated ligase enzyme then binds to DNA with nick. Adenosine monophosphate is transferred from ligase to 5' phosphoryl group of nick, and then the formation of phosphodiester bond takes place between 3' group of one DNA chain and 5' phosphate group of the another DNA chain. In addition to this, AMP is released. And now the DNA chains are ligated or sealed. Now let us discuss the mechanism of action of DNA ligase enzyme in detail. DNA ligase enzyme catalyzes the formation of phosphodiester bonds between 3' ends of one nucleotide, which is acceptor, with 5' phosphate end of another, which is donor. So this is the DNA ligase enzyme and it has lysine at the active site with free NH2 group at the side chain. Then binding of ATP takes place to the ligase enzyme, adenosine triphosphate. This bond breaks and then pyrophosphate or inorganic phosphate is released or removed. Then a covalent bond is formed between the free NH2 group at the active site and the AMP or adenosine monophosphate. This binding of the AMP to the free NH2 group is known as the adenylation, and this results in the activation of DNA ligase enzyme. Now the DNA with Nick comes and ligase enzyme binds to this DNA. This O minus, which is a nucleophile, attacks to the phosphate of AMP. This is known as the nucleophilic attack, as a result of which covalent bond between NH2 and AMP breaks, and now the AMP or the adenosine monophosphate is transferred to the 5-phosphate group of the DNA chain. So ligase comes to DNA with Nick, then takes place nucleophilic attack by oxygen O minus, which is nucleophile, of 5-phosphate group of Nick to AMP molecule, adenosine monophosphate molecule. This is known as adenyl transfer. So adenyl group is transferred to the 5-phosphate of DNA chain. Then H plus is removed from 3-OH group. Now the 3- end of the DNA chain has O minus, which is a nucleophile. Then the nucleophilic attack takes place by O minus to the 5-phosphate of the 5- end of DNA chain. This AMP is then removed and phosphodiester bond is formed between 5- end of one DNA chain and 3- end of the another DNA chain. This is known as Nick sealing. So here phosphodiester bond is formed and the Nick is sealed. So Nick sealing takes place by the formation of phosphodiester bond. This is all for today's video. In the next video, we will discuss about termination step in prokaryotic DNA replication. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.